Good afternoon. And so first, thank you to the organizer for the invitation. It's my pleasure to share with you our more recent effort to develop EV-based vector for drug-targeted delivery. So as you heard before, extracellular vesicles are natural vector for intercellular communication. And for a long time, they have been thought to be a very promising new class of vector for drug delivery. But the developments of such a type of vectors has been limited and delayed, mostly because we didn't know enough about how the machinery and how the cell work to really uptake the EVs and how the EV could deliver their content inside the acceptor cells. And I believe that my basic research laboratory contributed to fill this gap. So over the last 5 years, we basically characterize at the cellular, but also know at the molecular level, how EVs can deliver their content inside the cells. And I think we still partially a long time debate. So very briefly, the conclusion is that we formally demonstrated that EV can deliver their content inside the cell, but it's a low-yield bioprocess. And so therefore, our conclusion when we decided to contribute and participate to the development of EV-based vector, we concluded that the first task that needs to be done is to improve the efficiency of the EVs in terms of delivery capacity. And for us, the simplest way to do that was to control 3 important steps in the bioprocess: the loading of the cargo inside the vesicles, the targeting of the vesicles towards the cell of interest, and the final step, which is increasing the fusion between the membrane to increase the delivery capacity. So that's what we developed. It has been published, patented, and highlighted already by INSERM. And what I would like to do now in a few minutes is just give you the key results for each modular modules. So for the loading system, we took advantage of a well-established drug-inducible bioprocess, which enabled to force the loading of any peptide-biased cargo inside the vesicles. And as you can see on the graph on the right part, so when we introduce the dimerizer, we can very efficiently load any type of protein inside the vesicle. So the second module is what we call the fusion system to enhance the delivery capacity. And here, we basically repurpose a human physogen called syncytin-1 that is normally involved in placenta formation. And by doing that, we could largely increase the delivery efficiency and reach, as you can see on the graph, same efficiencies that viral proteins that usually are used, but introduce many safety issues. So in other words, we could compete with viral components and ruling out the safety issue of using viral protein. So the last module is what we call the targeting system. And here we develop a very versatile system that really enables to recruit at the surface of the vesicle any type of validated biotinylated antibodies that can be for R&D grade or clinically approved. So the idea here is to recruit the antibody to target the cell toward the cell of interest that will harbor at the surface an antigen of interest. And as you can see here on the graph, you can see that the targeting is very efficient, and we can very precisely target, in that case here, CD8 positive cells. And you see a nice dependency of the dose when we increase the amount of CD8 positive cells. So now when we assemble these three modules, we decided to develop new application. Because we are very early stage, we call that application, but hopefully in the near future, they might become products. But we will need time for that and partners. So the first application that we targeted is killer EVs. So basically our goal was to develop killer EVs to eliminate tumor in the future. And so here we took advantage of our technology, and basically we loaded a very lethal toxin inside the vesicle to kill the acceptor cells. The second application that we developed are so-called editing EV. So here the goal is to load the editing machinery, Cas9 CRISPR, and basically be able to reprogram the cell of interest. And in our case here, we could already reprogram CD8 positive cells. Just some example with a more mature application, the killer EV application. So we completely validated the efficiency of the killer EVs in vitro. And here you have an example from our paper, when you can see in the orange histograms that very efficiently we can almost completely kill the acceptor cells. And now we move forward in vivo studies, and we just started to do in vivo validation by testing anti-tumor activities of the killer EV in mice bearing a human tumor. And as you can see here, we can see a reduction of the tumor progression, so that's the blue curve, and when we do multiple injection of EVs. So that's a work in progress, and of course we need to continue to see if we can stabilize and completely inhibit the tumor growth. But I think it's promising. So that's not the only activity. So what I presented here is basically what we believe will be the generation 1 of EV-based vector. But we believe that what needs to be accomplished to be even more efficient and close to the biology is to really discover the key protein that control the endogenous uptake of vesicles. So for that, our goal is to discover the key machinery that control the uptake and the control delivery. And for that, we are performing a genetic approach with a unique library that we call surfaceome, because we are targeting all the proteins that are at the entry point of the vesicles. And hopefully, in the upcoming years, we will know what are the endogenous proteins that are specifically involved in the uptake, and more importantly, in the fusion, to position us in a unique place to have a new generation of physiologically relevant EVs. So we presented a new technology that presented several advantages that I discussed already. Obviously, we're at a very early stage, and we need to establish partnership with a scientific partner and also sponsors to, at someday, hopefully position the EV-based vector in existing pipelines and in new pipelines that will hopefully enable to reprogram cell in vivo. Thank you for your attention.
Thank you.